So let's talk about repeats first today. So what are repeats? They are actually short or long patterns of nucleic acids. Either they are found in DNA or in RNA. So actually they are the similar sequences of nucleotide bases that are found in the nucleic acids. And repeats are divided into 4 types. First we have the tandem repeats. Then second we have the inverted repeats. Third we have mirror repeats. And 4 we have palindrome repeats. First let's discuss about the tandem repeats first. Let's talk about tandem repeats first. They are also called as direct repeats. They are found on the same single strand. For example, we have a strand of 5' to 3'. And we take an example of a nucleotide base ATC. If this ATC is repeated on the same strand, then we call it as direct repeats. For example, we have this ATC is repeated again, ATC is repeated again, ATC on the same strand. We call it as tandem repeats. They are actually found in satellite DNA. Secondly, we can discuss about mirror repeats. There are also repeats that are found on the same strand, but they should be mirrored without any complementation. Meaning, the mirror images of ATCG should be found on the same strand, and if they are found on the same strand, then we call it as mirror repeats. For example, we have this ATCG. If we take the mirror images of ATCG, we get GCTA. And if these two are found on the same strand, we call it as mirror repeats. And for palindrome repeats, we have the repeats that are found on different strands, but they should be mirrored. Meaning, if the mirror images of ATGC, which is CGTA, if CGTA is found on the different strand, not on the same strand, but on the different strand, we call it as palindrome repeats. Here, have a look at this. We have two strands. From the upper strand goes from 5' to 3'. The lower strand 3' to 5'. Okay. So if we take the mirror images of ATGC, we have taken this nucleotide sequences as an example. If we take the mirror images of ATGC, we get ATGC, mirror images of ATGC would be CGTA, and if CGTA is formed on different strand, then we call it palindrome repeats. Meaning, the mirror images of ATGC, which is CGTA, is found on different strands rather than on the same strand with ATGC. We call it as palindrome repeats. Here, have a look at this. ATGC, CGTA, but CGTA is found on the lower strand, but ATGC is found on the upper strand. This type of repeats we call it as palindrome repeats. Also, we can check easily by reading from 5' to 3'. Same way on the lower strand if we read from 5' to 3'. So if we read the upper strand from 5' to 3', we get ATGC. And if we read same in the lower strand from 5' to 3', we get ATGC. Then we call it as palindrome repeats. Note that it should be mirrored on different strands. Next, we have inverted repeats. They are the repeats that are found on the same strand, but they should be mirrored and complementary to one another. For complementary, we have to know base pairing. For complementary base pairing, we know that A complements with T, and C complements and base pairs with G. For example, if we take a single strand 5' to 3', we take a nucleotide sequence of ATCG For the mirror images complementation of ATCZ, we have CZAT. Here, have a look at this. ATCZ. The mirror images would be CGTA without any complementation. But if we take the complementation mirror images of ATCZ, we get for G we have C, for C we have Z, for T we have A, and for A we have T. So the mirror complementation of ATCZ would be CGAT. And if these two are found on the same strand, then we call it as inverted repeats. Noted it should be on the same strand but mirrored and complemented to one another. They are actually found in transposons or jumping genes. For the summary, we have repeats, and repeats are divided into 4 types: tandem repeats, inverted repeats, mirror repeats, and palindrome repeats. Tandem repeats are the repeats that are found on the same strand and if the same sequences are repeated. For example, we have a strand of 5' to 3', and if we take a sequence of ATC, and if this ATC is repeated on the same strand, then we call it as tandem repeats. For example, we have ATC repeated again, ATC repeated again, ATC on the same strand, we call it as tandem repeats. For the inverted repeats, they are also repeats. They are found on the same strand, but noted that it should be mirrored and complementary. For example, we have a strand of 5' to 3', and we take a nucleotide sequences of ATCZ, and if we take the mirrored complementary of ATCZ, we get CZAT. And if these two are found on the same strand, we call it as inverted repeats. And for the mirror repeats, we have the repeats that are found on the same strand, but noted that it should be mirrored without any complementation, meaning it should be complementary. For example, we have a strand 5' to 3'. If we take sequences ATCZ, if we take the mirror images without any complementation of ATCZ, we get ZCTA. And if these two are found on the same strand, we call it as mirror repeats. And for palindrome repeats, we have the repeats that are found on different strands, but noted that the repeats should be mirrored without any complementation. They are actually the mirror repeats. Palindrome repeats are actually the mirror repeats, but the repeats should be found on different strands. For example, we have 2 strands 5' to 3', and the lower strand is 3' to 5'. And we take a nucleotide sequences of ATCZ on the upper strand, and if we take the mirror images without any complementation of ATCZ, we get ATCZ without any complementation mirror images of ATCZ would be ZCTA, and if ZCTA is found on different strand, then we call it as palindrome repeats. Here ZCTA is found on the lower strand and ATCZ is found on the upper strand. We call it as palindrome repeats. They are actually noted that they are the mirror repeats. They are found on different strands.